So our last speaker this morning, last but certainly not least, is Professor Phil Jager, who's eminent. He's published so many great pieces of work contributing to dementia. I should also put out a good shout out because he's also an old friend and colleague of our co-director, Adrian Iverson, back from their Harvard days. So it's a real great pleasure to welcome Phil, who's also got the coffee award, because of course he's the other way. He's actually kind of 7, 8 in the morning over there in the States. So Phil, thank you ever so much for joining us. And over to you. I should just say, sorry, the title of your talk, Defining Cellular Communities in the Aging Neocortex and the Relation to Neuropathological and Clinical Indices. Thanks ever so much, Phil.
Of course, thank you, Caleb, for a great introduction. And I have to admit, I missed the first session, which was a little bit early for me, but again, a terrific symposium, and we're really happy to be part of it. And thanks for inviting me. So yes, I'll be sharing with you some of our work based on the single cell data that we've generated so far, and there's a lot more coming, but this one I think is a little bit of a preview for what will be coming. So here are my disclosures briefly, and so maybe to take a step back, what we're trying to do on our end is to try to really understand the molecular state of the aging brain, and the challenges, as shown here to the right, there's a plethora of different forces that are acting on the molecular state of the brain. Of course, there's the whole development of the brain in the first place, but then life experiences come in. The systemic immune state is affecting things, and there are also positive effects like brain reserve and resilience, and confounders like biological rhythms that we've annotated over the years. So there's really a large number of different vectors that are affecting the molecular state of the brain. Now, how can we begin to really model this fully? And of course, we have to work at multiple scales. And I think this is one key point, I think, from this symposium, where we're beginning to look at more deeply at the single cell level. But I think, again, there are different pieces of information that we can grab from different dimensions of information. And of course, histology has for a long time provided single cell level data, although at much lower dimension. And I think on our end here at Colombia, what we're trying to do is to really have a programmatic approach to this, and to really sort of integrate not only the tissue-based work that I think we've talked a lot about today, but also the antemortem context. Because of course, when we're looking at the postmortem sample, this is really later in life. Typically, people are now dying at an advanced stage. And what we really care about in terms of the clinical context is, of course, much earlier. People in their 50s and 60s, this is where the interventions will have the most effect. And the big question, of course, is how does what we measure in these older brains relate to what was happening 20, 30 years before? And so this is where we have to integrate other types of data like in vivo imaging in humans and spinal cell, single cell RNA-seq, which I think is coming of age. And I think there'll be a lot more publications coming out this year. And again, maybe to give one sort of a set of vignettes to sort of illustrate, what we're trying to do here, that with multi-scale modeling, we've been working at the bulk tissue level for quite some time. This is just one example where we identified modules of co-expressed genes, which are shown here in purple on the left. The different modules, there are 5 modules that relate to microglial transcriptional activity. And what we're able to do is to resolve which modules were associated with different forms of pathology. In this case, we associated 2 of them with amyloid pathology, and a different one, module number 5, with tau pathology. And what's interesting here is that module 5 not only, so we have a set of genes that are found in the cortex that are expressed in microglia, and we found they were also, this module was also associated with a proportion of activated microglia defined morphologically, so these stage 3 microglia. But of course, the big question is, well, does this module really represent a separate cell type, or is it simply a transcriptional program that is found in the multiple different types of microglia? And of course, we couldn't resolve this at the bulk level. This ties in with, of course, a lot of histological studies, which have looked at these stage 3 microglia over the years. This is our effort from a few years ago, where we show that the proportion of these stage 3 microglia, activated microglia, is associated with a variety of AD-related traits, including both pathology and cognition, and where our modeling suggests that they are important, or they may play a role in the accumulation of tau pathology independent of amyloid, although there's some interconnection between the two, and that also, and tau, of course, causes cognitive decline. But again, here we have cells defined morphologically, and what is actually the molecular state of these morphologically defined cells? So of course, now that we have single nucleus data, we can begin to try to to bridge this divide between the single cell and the molecular state. The cohorts that we work with primarily are two designed and run by David Bennett in Chicago, disorder study and the memory aging project, ROS-MAP for short. These are prospective cohorts of older individuals who are non-demented at study entry. And so unlike a lot of other studies, here we're looking really at not quite a population-based, but similar to a population-based sample of older people followed over time with a detailed antemortem trajectory of cognition, and then collection of the brain at the time of death. So we can of course do, compare people who have AD to those who don't, whether that's clinical or pathological status, but also a lot of quantitative traits, because of course we have an entire distribution in this sample of older people. And as these collections are well known, they have really a ton of different phenotypic data that I don't have time to get into. Over the past decade, we've been generating with David a large number of different high dimensional data, which are listed here at the top right. And all of the data is available through the AD Knowledge Portal, which was created thanks to the Accelerated Medicine's Partnership for Alzheimer's Disease and NIA, the National Institute of Aging in the US program, to help disseminate and harmonize high dimensional data. And again, so our recent single nucleus data is already there, and more will be coming soon. And again, as I mentioned, there's a lot of different traits. I'm only going to talk about some of them today, but there's a lot more behind that. So as far as this initial work, it's reported actually in a bioRxiv paper from earlier this year. This is a pilot of 24 individuals that we did a few years ago. And we've now completed sequencing on 500 participants in ROS-MAP. So there we're currently finishing the QC of those data, and they will become available later this year through the AD Knowledge Portal. These participants, as we talked about earlier, we took several years to QC these data, again, as methods improved over time, and also both on the experimental side and the analytical side. At the end, we actually had a large number of cells, 170,000 transcriptomes, again, and consistent with what other people have reported today. I think we're producing a lot more data from each sample and also better data. This initial set was composed of 4 groups that represented different states of individuals. So we had individuals who were either cognitively non-impaired or who had AD dementia, so the clinical syndrome, and then who either did or did not have a pathological diagnosis of Alzheimer's disease. So again, maybe a reference group that is, you can call these control subjects, resilient, vulnerable people, and then people who had both a diagnosis of AD dementia and a pathological AD. And then again, we did sort of a standard single nucleus sort of sequencing of these individuals. Although, again, we spent a fair amount of time optimizing our pipeline. What we've reported so far is, again, very consistent. We defined the major cell groups that we heard about earlier that are shown here at the top left. And as other people have reported, the proportion of neurons is relatively homogeneous across individuals. And the glial cells are, of course, much more dynamic across different subtypes. And then if you look at men and women and also the different categories of subjects that we have, we do see sort of large differences in the number of cells that may be present in one or the other of these groups, which is shown here in these density plots. But because we had a much larger number of cells than earlier efforts, and also higher quality data, we're able to resolve more subtypes of cells than have been reported so far, although again, this is rapidly changing. And what we described are a number of different subtypes for astrocytes, microglia, endothelial cells, and neurons. And I think an important theme that I would like to sort of share is that while we were able to resolve subtypes for these cells, there's also an argument to be made that maybe the subtyping them or clustering them, it may not be the best approach because they're quite homogeneous in many ways. And sometimes the boundaries for those cell types made sense. But when it came to oligodendrocytes, making a hard boundary really didn't seem to make much sense. And so instead, we followed a topic modeling approach here to really identify the major axes of variation in the data, and then to use those as the driver for downstream analyses. So again, I think what we'll see going forward is that there's probably not a one-size-fits-all approach to analysis of different cell types. You really have to tweak and sort of select the best method for your cell type of interest. Another point that we were able to make, since we have separate efforts to sort live microglial cells from human autopsy and surgical tissue, this was published last year. We compared our data from the single nuke data, which was about 4,000 microglial nuclei, compared to the 16,000 live microglial nuclei we have collected separately. I think the important point here is that we were able to find the same types of cells in both data sets. So I think very reassuringly, we don't seem to be missing major sort of subtypes of microglia between the two approaches, whether the nuclear approach on frozen material versus the fresh tissue that's sorted for live cells. Of course, the live cells, we have more cells, we have more data, also there's better quality data because these are live cells. And so we're able to resolve, as you can see it in the little heat map, we have more subtypes from our paper of different microglial states compared to the nuclei-derived clusters. But you can see here that they really align quite well between the two. And I think as we get more nuclear data, we'll be able to sort of make what we did with the live cells. But again, I think reassuringly, both are present. Also, some of these, there are differences, of course, in the level of expression of different genes, but what we see is that while the level of expression may be different in frozen tissue versus live cells because of the processing differences, of course, the cells on the genes are not absent, but there are some differences in expression. Another point that we were able to really make here is that there's extensive interindividual variation among the non-neuronal cell types. And again, other people have reported this as well. But I think what's very important is shown here, particularly for astrocytes at the top left. So what we're showing here are the 5 astrocyte subtypes that we've identified in columns. And then in rows, we have the individual subjects, our 24 subjects. And then what we've shown in the color is basically the proportion of the astrocytes that are assigned to a given state for a given person, with the orange color denoting the high frequency. And you can see that for most individuals, astrocyte cluster number 1 is the dominant cluster for these cells. And this makes sense because these are what we think are the homeostatic astrocytes. But what you can see is that where some individuals have the dominant subtype being not astrocyte 1, but astrocyte 2 or 3. And this is only a minority of people. So I think unlike other cells, like you can see the microglia, the oligodendrocytes here, and the epithelial cells, it's really sort of more abrupt in the astrocytes, where when individuals do have these other states, and everybody has these states, but in most people, they're quite minor populations. But in some people, there's an enormous flux over to these altered states, and they become the dominant composition of for the astrocytes. In terms of individual variation, again, we noted some interesting observations. For example, there are 2 of our inhibitory subtypes are strongly anti-correlated, as you can see here in the middle, this inhibitory 5 subtype, inhibitory 3. So there's a very strong correlation between the 2. And what's interesting, it's also related to its proportion of excitatory neurons. So as the proportion of excitatory neurons goes up, which is shown here at the bottom, you can see in blue, the proportion of inhibitory 5 neurons goes down while inhibitory 3 go up. So again, we're just beginning to note some of the different levels of heterogeneity that are existing in data structure that are existing in these data. Now, because we only had 24 individuals, and as was shown nicely by Alan, the sample size is really not adequate to do any sort of meaningful statistics. And so, we didn't pursue that in the 24 subjects. But what we, of course, had was bulk data on 640 subjects. So Vilas Menon, my colleague here at Colombia, develop a new algorithm, which he calls a cell-mod, where we trained the algorithm on 24 individuals, which had both the bulk RNA-seq data and the single nucleus RNA-seq data from the same brain region. And so based on these individuals, we were able to infer the proportion, not just of the major cell types, but all of the different cell subtypes that we have as well. And what we validated that, you can see that, again, the correlation is not perfect, of course, because this is bulk data, but the correlation is reasonable. And you can see on the right that there's a correlation also with the proportion of these cell types measured by immunohistochemistry in the same individuals, although in the other hemisphere. So that's part of the lower correlation, perhaps. Now, with these data, we can then begin to look at things at a more appropriate scale for statistics. Now, what we had also noted in our single nucleus data in the 24 subject, which is shown here on the left, you can see that the proportion of certain cell types is more correlated to one another and anti-correlated to some of the other cell types. This is at the high level. When you go down to, on the right, you can see here the 24 individuals. If you look at all the different cell subtypes that we've enumerated, you can see that there's a lot of structure. A lot of these different subtypes are correlated in frequency with one another. And we can see that in our inferred proportion, in our much larger data set of 640 individuals, this correlation structure exists as well, and is relatively conserved. And so using these correlation sort of maps, we were able to create and define these, what we call communities of different cell subtypes, whose frequency are correlated with one another. And one example here is shown on the left. So this is the model that derives from the actual single nucleus data in 24 individuals. And we have, for example, one community here at the top left, which is composed of the astrocyte 1, endothelial 1, and microglial 1 subtypes. And these are all the homeostatic cells and their frequency is correlated. Again, intuitively, this sort of makes sense. And it's actually inversely correlated to this other population here at the bottom, which is composed of microglial state 2, astrocytes state 2 and 3, which have aspects of activated cells. Again, so at a sort of very simple level, this seems to make sense, but we're beginning to see that there are different cell subtypes that are correlated frequency to one another. Now, whether this represents topologically defined communities or cells that are more distributed but responding to similar signals, we don't know, of course. We will have to look into some of the emerging spatial transcriptomic data that we have to try to address this point. But excitingly, this structure was preserved in the 640 individuals with inferred cell subtype proportions. And so this is shown on the right over here. Then another thing we can do is to, of course, look at since we have this large data set, now we can begin to look properly at association with different traits. And so we see here that we can do association of the frequency of a given cell subtype with different traits. Shown on the left is the association with beta amyloid on the y-axis and cognitive decline on the x-axis. And you can see here that there's a number of different associations. Some of them are consistent for amyloid and cognition, for example, this group at the top left quadrant. But you can see that there are a lot of changes that are related to amyloid beta that are not correlated with cognitive decline at all. So these are the black dots in the middle. If you look, on the other hand, at the middle graph, you see that if you look at the tangles and cognitive decline, the correlation between the two sets of results is much stronger. Again, consistent with the fact that tau pathology accumulation, or the burden of tau pathology, is much more strongly correlated with cognitive decline than amyloid pathology. And if you compare the two pathologies on the right, you can again see that while there are some that are consistent across the two, for the most part, amyloid really causes a large change in cell frequency, but they don't seem to be really strongly correlated with changes in the accumulation of tau pathology. Now, for some of the later modeling that will work, I'll sort of direct your attention to some of these oligodendrocyte and inhibitory neuron frequency, which were really the ones that had the most extreme p-values in our analysis. The other thing we can do is to map these association results back onto our little network of our communities. And what was interesting here, if we show the results for cognitive decline, and we've colored the cell types that are significantly associated, and in gray are the ones that are not associated with cognitive decline. you can see that there is a little cluster here, which clearly they're all interconnected and correlated in frequency with one another. So you really seem to have a community of cells that is being altered, as a group in the context of cognitive decline. And the similar one is associated with tau, which is shown here in the middle. And again, you can see that for the amyloid beta, while there is some similarity, there's a lot more changes going on in more neurons, especially the excitatory neuron populations are all more altered in frequency in relation to amyloid beta deposition. Finally, we can do some modeling, because of course we would like to know, well, are these are associations, but so can we try to infer the directionality of results? And of course, we can't formally talk about causality. These are simply models, but they're hypotheses that can then be tested. And so using Mendelian randomization, where we know that APOE4, like genetic risk in general, comes before everything else, so we can do some modeling there, which suggests that the changes in oligodendrocyte 3 population, inhibitory 5, the most extreme results in our associations, seem to be coming after the accumulation of tau pathology. And more specifically, here at the bottom, you can see that if we go one step further and look at cognition, both of these changes, whether it's the proportion of OLIG3 cells or the proportion of inhibitory 5 cells, they are occurring downstream of tau, but upstream of cognition. They're explaining some of the effect of tau, although not only a proportion of it, on cognition. And so what we're looking at, I think, here are changes that are occurring as a result of tau pathology and ultimately are contributing to a decline in cognition downstream. So for the summary of these single nucleate data, I just wanted to sort of quickly summarize. Again, we have novel subsets of these different glial cells. We have high resolution with our data. Again, I think this is a rapidly evolving field, and we're looking forward to more data from our own group, of course, and all the other groups to sort of improve on this. I think an important point I mentioned was that the minority of individuals have a large number of some subtypes of astrocytes, for example. So there's a lot of heterogeneity in a minority of individuals. And the CellMod method works not just on the ROS-map data, but on any data set. So that's exportable as well. So again, in case people are interested. And then again, we define these as cellular communities, which are mixed neuroglial in composition. And of course, the big question is whether these represent topological groups or not. Now, in the last few minutes, I just wanted to give a little bit of a preview of a related project. Again, this is our microglial sort of effort in the live cells that are extracted from fresh autopsy tissue, and basically expanding on the paper that we published last year. So again, this is the paper that was published. I'm not going to go through this, but we have basically a process to sort these cells and that are alive, and then to sequence them. Initially, we presented a model with 9 different subtypes of microglia. I think the important component of that paper was that there was not, again, as other people have also reported, in humans, the context is a little bit more complex, and there's not a single disease-associated microglia. That signature is more distributed in humans. And so we've now expanded this effort to more subjects. The current frozen version of the data has 75 subjects, up from 16. And instead of 16,000 transcriptomes, we now have 215,000 transcriptomes. So we have a lot more data. And also, as you can see on the left, we have a much wider spectrum of individuals with ALS, Parkinson's, and other diseases beyond, and more Alzheimer-related subjects as well. So I think with this, we're trying to improve on our earlier model and to resolve, smaller subtypes potentially, which we have done. And again, now we go from 9 to 13 different clusters of microglia. In general, actually, all individuals have these clusters. So while there is some difference in frequency for different states, particularly ALS seems to be quite polarized in some ways. The microglia are not unique to a particular subtype of disease, and the majority of the cells in the tissue, even in disease, are still homeostatic microglia. And again, what's shown here is that the homeostatic state is actually refined into 2 different polar types of groups, with an intermediate homeostatic step between the 2, and then there's all the other groups sort of organized around the central axis of differentiation of homeostasis. And of course, we can do different things with this. For example, we can look at, given that I'm a clinical neuroimmunologist, I'm very interested in MS, and we can see here that there's an enrichment for MS susceptibility genes really on one end of this of the main axis of variation for microglia, which is shown here. And then we can also use our data to model other questions, like we've now done single cell sequencing in CSF, and we can see here that there are microglia-like cells in the CSF, although we don't think they're true microglia. I think we have some data we're working on to address that question. What we can see on the right is if we take the microglia-like cells from the spinal fluid, and then we look at what type of microglia do they look like from our brain atlas, it's really quite different. So in blue, we have the proportion of different microglia from the different states in the brain, and in red are the results from the CSF. And you can see that in CSF, it's really clusters 6 and 7, which are enriched in CSF. And those happen to be the cell types that were at that polar end in the MS analysis, because these happen to be CSF samples from MS and other inflammatory diseases. So with this, again, we have a programmatic approach to multiscale modeling. We've made a big investment in a single cell characterization that is sort of beginning to bear fruit, and a lot more data is coming out this year. Importantly, at least for microglia, the live microglia and the frozen nuclei seem to be yielding consistent data, which is exciting. So I think we'll be able to go back and forth between these two different types of data. Importantly, also for some cell types, like all the oligodendrocytes in this approach, we didn't use a clustering approach. We more of a topic approach. And maybe I'll sort of stop there given the time. And I'll sort of thank my colleagues, particularly Vilas Menon, who's a computational biologist in my center, and who's played a key role here, as well as Naomi Habib at the University of Jerusalem. And we've worked together on the single nuke data. Thank you.
Fantastic. As always on Zoom, you can't hear the audience clapping, but they're clapping away. It's incredible work. So, I mean, one of the things that I'll just say, everyone, please put your questions into the Q&A, and I'll read them out, or panelists into the discussion, the comments field. But one of the things that really came out, and you're seeing these interindividual differences between particularly for the glia. One of the things that came out from the first studies, for example, from the Broad, was this difference in gender for the microglia. Do you see how big a factor is gender in differences that you can see in your microglia populations?
Yeah, in we certainly do see differences in expression related to gender, and we're currently analyzing that in our larger microglial data set from the live microglia. In terms of the frequencies, we don't see huge differences, but again, I think it's a little bit confounded because we have different diseases. So it's a complex data set to try to answer some of these questions. But again, in a couple of years, we'll have a couple hundred individuals, hopefully, processed, and I think we'll be better able to address some of these questions. But I think as we do our analyses and report on it later this summer, we should be able to say something about gender, but I think those are still in progress at this point.
Fantastic. And now you're getting these particularly distinct subtypes. Are you able to associate one a subtype or the other, particularly with genetic risk from when you're in your control brains? So when you intersect them with genetic risk using, I don't know, L-score regression or magma or whatever it is, are you highlighting one particular population as being perhaps a likely more causal to contributor?
Yes, I think so those analyses are still sort of underway. I shared with you some of the results from our MS, sort of work, for example. And again, I think causality is always a difficult sort of question to address with cross-sectional data. But I think we will be able to make some progress, I think, to try prioritize subtypes. And I was a little surprised, actually, in the MS results, for example, that we had seen earlier in enrichment more generally in microglia. So I had expected a more distributed, enrichment of the MS genes. But here, I was quite surprised that there is really just one aspect of this cloud of microglia that is really strongly enriched. So it really suggests that there are some transcriptional programs specific to a subtype of microglia that are going to be more involved, at least for that particular disease. Again, we have to do the same for Alzheimer and other diseases.
I suppose, I mean, I suppose my last question would, there's been discussion, and I saw some kind of papers suggesting that that actually there was some transitions between cell types, you know, for example, with Priscillin 1 and so forth. And I'm you know, these are early days. I haven't seen too much in it, but if you're talking about the fact that you are seeing big differences between individuals in these particular populations, and perhaps some of these populations are more associated with a predisposition, do you think that actually that you're capturing a static state, that people are kind of, if you like, have a predisposition to have a certain frequencies of population, or those populations are very dynamically changing over the course of people's lives, and that could be influencing disease?
Mm-hmm. No, those are great questions, and I think we will be able to address them once we look at our data set from 500 brains. I think there we'll have the power to look at this properly. So far, we don't know. I would have to say, you know, all we see is that at the time of death, you know, there's sort of this big difference. And I think, again, what's going to be key to address your question is, of course, longitudinal data. And so this is where, you know, we're working with some colleagues here on the, you know, sort of third generation TSPO ligands to do longitudinal PET imaging in individuals, and also for spinal fluid sampling would be key, you know, to address some of these questions in terms of stability over time.
Yeah. No, it's great questions. Brilliant. I've just asked apparently the questions that came through in chat, so we've got those. So brilliant. So we thank you ever so much, Phil. Thank you for joining us. We know it's very early hours now, and please do stick around because we're really looking forward to chatting to you in the workshops later. So thank you ever so much to our speakers in this last session.